Hey there, I am Maruk Said, a microbiologist and a science educator. Science is my passion and I'm here to share it with you. Virology topic on Nipah virus. But before starting this video, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of the videos. Let's begin with the table of contents. First, we will have a brief introduction of Nipah virus. We will learn its structure and genome structure, its epidemiology and transmission methods, how it replicates, pathogenesis of Nipah virus, and what are the clinical manifestations, its diagnosis, treatment, and lastly, prevention and control. Let's begin with the introduction of Nipah virus. The paramyxovirus include the zoonotic Nipah virus, which is transmitted by bats. In Malaysia, it was originally discovered in 1998. Since 1999, however, no instances have been found in Malaysia. Numerous epidemics in South and Southeast Asia are brought on by the virus. The primary reservoir for the infection from which illnesses in people and pigs can spread is Theropus fruit bats. The Malaysia-Singapore outbreak of the virus was associated with contact with pigs, whereas the Indo-Bangladesh outbreak was associated with consumption of raw date palm sap contaminated by fruit bats. The virus, which is extremely contagious and spread through infected people and animals, causes severe neurological and respiratory illnesses. The virus is categorized as a Biosafety Level 4 pathogen because of its high mortality rate, rapid rate transmission, and lack of an effective antiviral drug or vaccine. Now let's learn about the structure of Nipah virus. The paramyxovirus family of enveloped viruses include the Nipah virus. It has a helical nucleocapsid and a negative sense, single-stranded, non-segmented RNA genome. The viral ribonucleoprotein is made up of a long polymerase, phosphoprotein, and nucleocapsid. The matrix protein that surrounds the nucleocapsid is embedded with spike-like fusion proteins and glycoproteins that are in charge of cellular adhesion and host cell interns. The Nipah virus range in size from 40 to 1900 nanometer, making them bigger than ordinary paramyxovirus viruses on average. The presence of reticular cytoplasmic inclusions near to the endoplasmic reticulum distinguishes the virus from other paramyxoviruses. The virus closely resembles the Hendra virus, differing just slightly in its ultrastructure and exhibits strong cross-reactivity in a number of serological assays. The two strains, Bangladesh and Malaysians, vary in their virulence and mode of transmission yet have a 92% sequence similarity. If this video is helpful, don't forget to subscribe. Now let's start with the genome structure of Nipah virus. A negative sense, single-stranded, non-segmented RNA with a genome size of around 18.2 kilobase pair makes up the Nipah virus's genome. The nuclear protein, phosphoprotein, matrix protein, fusion protein, attachment glycoprotein, and the large protein or RNA polymerase protein are the 6 structural proteins that it codes for. 3 additional non-structural proteins are also encoded by the ping gene, either by RNA editing or an alternative open reading frame. Epidemiology of Nipah virus. In Malaysia and Singapore, the first Nipah virus outbreak was documented between 1998 and 1999. The diagnosis of severe viral encephalitis in 94 people who had intimate contact with the swine population between September 1998 and June 1999 demonstrated the virus's direct transfer from pigs to humans. Between 1998 and 1999, 246 patients in Singapore and Malaysia were diagnosed with febrile encephalitis caused by Nipah virus. The mortality rate for people was about 40%. In 2001, a geographical disjoint area, the Meherpur region of Bangladesh and Siliguri city of West Bengal, India, reported the second epidemic. In all, 9 Nipah outbreaks were documented in Bangladesh up until the year 2010. A further epidemic in Bangladesh in 2011 was reported to have resulted in a total of 15 fatalities from infection with the Nipah virus. Epidemiological studies revealed that the Nipah virus was spreading by zoonotic and human-to-human transmission throughout Asia, Africa, and South Pacific Ocean. Over the last 20 years, it has caused the lives of hundreds of people and constituted a serious threat to both people and domestic animals. Now let's learn about the transmission of Nipah virus. The Nipah virus can be transmitted to people from direct contact with sick animals such as pigs or bats, or their bodily fluids such as blood, urine, saliva, etc. Consumption of tainted food items that have been polluted by diseased animals such as palm sap or fruit that has been tainted by fruit bats. Close contact with the Nipah virus positive individual or their bodily fluids such as blood, urine, nasal or respiratory droplets. Human infections during the initial Malaysia-Singapore outbreak were from coming in touch with infected pigs or their bodily fluids. The intake of fruits and fruit products such as raw date palm juice contaminated with the saliva from the infected fruit bats was discovered to be the most likely cause of infection during the epidemics that followed in Bangladesh and India. The Nipah virus was shown to directly transfer from person to person during later epidemics in Bangladesh and India through close contact as well as bodily fluids and secretions. Numerous family members and carers of the afflicted individuals also reported human-to-human transfers. If this video is helpful, don't forget to subscribe. Replication of Nipah virus. Attachment and adsorption. The viral glycoprotein G and the B2 receptor help the virus bind to its host cell, which is then followed by the fusion of viral and host membranes. Penetration and biosynthesis. The transcription of viral messenger RNA, which is then translated into viral proteins, uses the genome of the negative RNA as a template. Additionally, the synthesis of cRNA plus uses the vRNA as a template, and C plus in turn uses the vRNA as a template. Additionally, the viral protein contributes to interferon signaling. Additionally, the cell endocytose and mature the F proteins. Assembly. N, P, C, M, F, and G are all included in the viron during assembly, which is predominantly carried by the M protein. Release. The virons are expelled from the cell after budding through the host membrane, which has G proteins on its surface. Let's learn about its pathogenesis. Number 1. In the initial stages of Nipah infection, the virus can be observed in epithelial cells of the bronchioles. Number 2. Nipah virus antigen can be identified in both the bronchi and alveoli. Number 3. Infection of the airway epithelium triggers the activation of inflammatory mediators. Number 4. As the disease progresses, the virus spreads to the endothelial cells of the lungs. Number 5. The virus enters the bloodstream, disseminating either freely or bound to host leukocytes, ultimately reaching the brain, spleen, and kidneys. Two distinct pathways facilitate viral entry into the central nervous system. One from the hematogenous route and the other interrogately with olfactory nerves. Infection of the central nervous system disrupts the blood-brain barrier, leading to the expression of interleukin-1 beta and tumor necrosis factor alpha, resulting in the development of neurological symptoms as indicated in red font for human symptoms. So, to summarize it, through the oral nasal pathway, the Nipah virus infects the host and spreads. It is still unclear exactly where the human first replication occurs. However, because of the large amounts of antigen present there after infection, the lymphoid and pulmonary tissues are regarded as potential first replication sites. Acute respiratory distress syndromes like conditions can be brought on by the viral antigens that can be brought in the bronchi and alveoli. These cytokinesis cause inflammation. The virus subsequently spreads to the lungs endothelial cells in the later stages of the illness. The virus spreads as a result of early viremia, and then the endothelium becomes the site of secondary replication. The eprin B2 receptor on the endothelium as well as on the lungs, placenta, prostate, blood vessels and several other organs is where the viral glycoprotein G binds. Eprin B2 is highly conserved across multiple animal groups with similarities between bats and pigs of about 95 to 96%, which helps to explain the great variety of hosts connected to the Nipah virus. The virus enters the brain either retrogradely through the olfactory neurons or hematogeneously through the bloodstream. It produces interleukin-1 and tumor necrosis factor which causes neurological symptoms throughout the infection. It also disrupts the blood-brain barrier. Because the virus avoids the innate immune response, it is very deadly. Interferon activity is also inhibited by the P gene products. The various Nipah virus strains are thought to have co-evolved independently with their reservoirs, which accounts for the variation in virulence and epidemiological characteristics. If this video is helpful, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my videos. Now let's begin with the clinical manifestation of Nipah virus. Infection with the Nipah virus can result in mild to severe symptoms including encephalitis and eventual death. The virus typically takes 4 to 14 days to fully incubate. However, a 45-day incubation time has also been recorded as being prolonged. General signs of infection throughout the early stages include fever, headache, cough, sore throat, difficulty in breathing, malaria, and vomiting. Severe symptoms might then develop, and within 24 to 48 hours, a coma could be developed. These signs might be disorientation, drowsiness, confusion, seizures, coma, brain swelling, encephalitis. Patients who have aberrant doll's eye reflexes, pulmonary reflexes, vasomotor abnormalities, seizures, and myoclonic jerks are said to have brainstem dysfunction. Relapse or late onset encephalitis, some of which can happen months or years after the initial infection, are characteristics of Nipah virus. Some individuals also have psychological symptoms such as sadness, personality problems, trouble concentrating, and memory loss. During the outbreaks in Malaysia and India, difference in clinical symptoms were also seen between the various strains. In contrast to the outbreak in Malaysia, where no significant respiratory involvement was seen and had a relatively low mortality rate of 40%, respiratory illness like cough, respiratory distress, and atypical pneumonia were observed in 70% of patients there. This outbreak in India and Bangladesh also had a higher mortality rate of 70%. Let's learn how Nipah virus is diagnosed. The samples for the serological testing, which may include throat swabs, urine, blood, and cerebrospinal fluid for diagnosis, should be obtained 10 to 14 days after the beginning. A BSL-4 laboratory should process the sample. However, following viral inactivation by sample irradiation, it may also be handled in BSL Lab 2. PCR. Real-time PCR can be used to identify Nipah virus RNA in respiratory secretions, urine, or central spinal fluid. High specificity and sensitivity characterize this method. The SYBR green-based test also detects the hepatitis E virus but only identifies a different portion of the N gene, whereas the Tagman probe-based assay detects the N gene and has a better sensitivity. Immunohistochemistry. Immunohistochemistry may be performed on a broad variety of formalin-fixed tissues from many organs, including the brain, lung, spleen, kidney, and lymph nodes. Previously, convalescent human serum was utilized for immunohistochemistry. Presently, Nipah virus-specific rabbit serum is used instead. Viral isolation. The Viro cell line is the preferred cell line for viral isolation from the samples, which must be carried out in BSL-4 facility. Within 3 days, cytotoxic effects are visible and Nipah virus formed Sixitia, which are bigger than those produced by hepatitis E virus and aid in differentiating the two are seen. ELISA. It is the most typical serological technique for finding Nipah virus infection. IgG and IgM are both detected with it. On the first day of illness, 50% of patients have IgM antibodies detectable, whereas 100% of the patients have IgG antibodies positive. IgG lasts for a number of months. Lastly, serum neutralization test. Although it needs to be carried out in BSL-4 laboratory, it is regarded as the most accurate test for detecting Nipah virus infection. The test sera are used to infect the Viro cells, which are then exposed to the virus for 3 days while being watched for cytopathic effects. Now let's see the treatments for Nipah virus. The Nipah infection can only be managed by supportive care and acute encephalitis treatments. Anticonvulsant medication, secondary infection care, mechanical breathing, and rehabilitation are some examples of supportive care. For the potential therapy of Nipah virus, 3 pharmaceuticals have been investigated: ribavirin, M02.4 monoclonal antibodies, and favipiravir. And lastly, prevention and control of Nipah virus. Number 1. An effective vaccine candidate against Nipah virus involves the use of a recombinant malicious virus vaccine engineered to express the enveloped glycoprotein of Nipah virus. Number 2. In recent years, a recombinant vaccine has been developed using a replication-competent vesicular stomatist virus which encodes the Nipah virus glycoprotein. Number 3. Vaccine research has yielded Nipah virus-like particles composed of 3 Nipah virus proteins G, F, and M produced from the mammalian cells. These Nipah virus-like particles have been successfully validated as vaccines in BALB sieve mice. Number 4. Cutting-edge immunoinformatic techniques have been harnessed to create a peptide-based Nipah virus vaccine. This innovative approach involves the prediction and modeling of T-cell epitopes from Nipah virus antigenic proteins. Prevention. The Nipah virus has few effective therapies, thus illness prevention must be given first emphasis. Despite the high mortality rates of one of the WHO's priority infections, there are still no licensed and effective vaccines for Nipah virus infection in humans. However, public health vaccines the University of Tokyo, and the University of Oxford are testing Nipah virus vaccines in preclinical studies with the intention of testing their safety and efficacy in the next years. Among the steps used to avoid the illnesses are interventions to stop the spread of virus to agricultural animals by keeping them from consuming fruit bat contaminated with the virus. Farms shouldn't be located next to the fruit trees that draw bats. Consuming tainted sap must be prevented. Installation of physical obstacles to stop bats from climbing trees. The specimens must be handled with minimal aerosol generation techniques in appropriate biosafety cabinets. Patients of farm animals suspected of having Nipah virus needs to be separated right away. Hospitals and healthcare facilities must be ready to screen patients and handle any outbreak in disease-prone areas. Proper hand washing and hygiene practice must be exercised before and after handling specimens or contact with the patient. So that's it for today. Thank you so much for watching till the end. Don't forget to like this video, subscribe to my channel and press the bell icon button.